Good day, this is the Professor, Professor CBMD PhD, and today's Wednesday, October 26, 2022. It is 6:21 and 15 seconds, a.m. Eastern Standard Time, and today's research, pancreatic cancer. So let's hop to it here. Gel-like radioactive tumor, implant obliterates, pancreatic cancer, in mice, combination of internal radiation and chemotherapy dissolves tumors in 80% of mice across multiple models. Date October 20th, 2022. Source, Duke University. The biomedical engineers have demonstrated the most effective treatment for pancreatic cancer in mouse models ever recorded. While most mouse trials consider simply halting growth a success, the new treatment completely eliminated tumors in 80% of mice across several model types, including those considered the most difficult to treat. The approach combines traditional chemotherapy drugs with a new method of irradiating the tumor rather than delivering radiation from an internal beam that travels through healthy tissue. The treatment implants radioactive iodine-131 directly into the tumor within a gel-like depot that protects healthy tissue and is absorbed by the body after the radiation fades away. Researchers did a deep dive over 1,100 treatments across preclinical models and never found results where the tumor shrank away and disappeared like theirs did. When the rest of the literature is saying what they are seeing doesn't happen, that is when they knew they had something extremely interesting. Despite accounting for only 3.2% of all cancer cases, pancreatic cancer is the third leading cause of cancer-related death. It is very difficult to treat because its tumors tend to develop aggressive genetic mutations that make it resistant to many drugs, and it is typically diagnosed very late when it has already spread to other sites in the body. The current leading treatment combines chemotherapy, which keeps cells in a stage of reproduction vulnerable to radiation for longer periods of time, with a beam of radiation targeted at the tumor. And this approach, however, is ineffective unless a certain threshold of radiation reaches the tumor. And despite recent advances in shaping and targeting radiation beams, that threshold is very difficult to reach without risking severe side effects. Another method researchers have tried involves implanting a radioactive sample encased in titanium directly within the tumor. But because titanium blocks all radiation other than gamma rays, which travel far outside the tumor, it can only remain within the body for a short period of time before damage to surrounding tissue begins to defeat the purpose. In all endless possibilities, there's just no way to treat pancreatic cancer right now. And to skirt these issues, researchers decided to try a similar implantation method using a substance made of elastic-like polypeptides, which are synthetic chains of amino acids bonded together to form a gel-like substance with tailored properties. Because elastic-like polypeptides are a focus of the research laboratory, they were able to work with colleagues to design a delivery system well suited for the task. The elastic-like polypeptides exist in a liquid state at room temperature but form a stable gel-like substance within the warmer human body. When injected into a tumor, along with a radioactive element, the elastic-like polypeptides form a small depot encasing radioactive atoms. In this case, the researchers decided to use iodine-131, a radioactive isotope of iodine, because doctors have used it widely in medical treatments for decades, and its biological effects are well understood. As the elastic-like polypeptide depot encases the iodine-31 and prevents it from leaking out into the body, the iodine-131 emits beta radiation, which penetrates the biogel and deposits almost all of its energy into the tumor without reaching the surrounding tissue. Over time, the elastic-like polypeptide depot degrades into its constituents, amino acids, and is absorbed into the body, but not before the iodine-131 has decayed into a harmless form of xenon. The beta radiation also improves the stability of the elastic-like polypeptide biogel that helps the depot last longer and only break down after the radiation is spent. There is also a new treatment in concert with paclitaxel, a commonly used chemotherapy drug to treat various mouse models of pancreatic cancer. They chose pancreatic cancer because of its infamy for being difficult to treat, hoping to show that their radioactive tumor implant creates synergistic effects with chemotherapy that relatively short-lived radiation beam does not. The researchers tested their approach on mice with cancers just under their skin by several different mutations known to occur in pancreatic cancer. They also tested it on mice that had tumors within the pancreas, which is much more difficult to treat. Overall, the test saw 100% response rate across all models, with the tumors being completely eliminated in 3/4 of the models, about 80% of the time. The test also revealed no immediately obvious side effects beyond what is caused by chemotherapy alone. Researchers say the constant radiation allows the drugs to interact with its effects more strongly than the external beam therapy allows. And that makes us think that this approach might actually work better than external beam therapy for many other cancers too. The approach, however, is still in its early preclinical stages and will not be available for human use anytime soon. The researchers say their next step is large animal trials, where they will need to show that the technique can be accurately done with the existing clinical tools and endoscopy techniques that doctors are already trained on. If successful, they look toward a phase 1 clinical trial in humans. The researchers have been working on developing new cancer treatments for close to 20 years, and this work is perhaps the most exciting they have done in terms of its potential impact, as late-stage pancreatic cancer is impossible to treat and is invariably fatal. The pancreatic cancer patients deserve better treatment options than are currently available, and researchers are deeply committed to taking this all the way to the clinic. Good day, this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.